All right, hello. Welcome to PacBio's first CoLab presentation of ASHG 2022. My name is Edd Lee. I'm the Director of Human Genomics Segment Marketing at PacBio, and I'm here to start us off with a quick presentation before I introduce our main speaker, Fritz Sedlazeck of the Baylor College of Medicine Human Genome Sequencing Center. Fritz will speak about challenging medically relevant genes, aka dark regions, and how to resolve them with HiFi and target enrichment. Today, my presentation, really quick before, will be focused on how we can help you unlock the next wave of discoveries in human genomics with PacBio technologies. So as far as new tools that we have available to help you unlock the next wave of discoveries, maybe we could just take a little walk down memory lane. Back in 2011, PacBio launched the RS system. It was the size of a minibus. It had about 150,000 zero-mode waveguides. 4 years later, in 2015, they launched the Sequel system, increasing the throughput by about 7 times, and they also shrunk the form factor to something that actually could fit in any lab. Another 4 years after that, in 2019, the Sequel 2 system came, and along with that came an 8 times increase in throughput. And then, as you may have heard, as of last night, the Revio system is now launching, which is our new high-throughput long-read system. And in 2023, in the first quarter, it will start to ship. And so 4 years after 2019 was the next innovation. Revio will increase the throughput of Sequel 2e by 15 times. So what kind of sequencing is coming off the Sequel and the Revio system? We call it HiFi sequencing or HiFi reads. So let me just quickly define what those reads are or what that sequencing is. HiFi reads are long reads, so the average read length is about 15 kb. They offer very, very high accuracy, so a consensus 30x whole genome would have quality reads of a quality score of Q50. They are single molecule reads, so they offer single molecule resolution, and each of the reads has an accuracy of, or the majority of the reads, 90% of the reads are over Q30. They also offer uniform coverage, so they have because they have the least GC bias, and so you can kind of resolve GC-rich areas. And then finally, they offer simultaneous 5MC detection. So with, not only are you getting A, C, G, and T, you're also getting 5MC. We call that 5-base sequencing, and it does not require any other types of library preps. It's all native to a single library prep. So what kind of research is being done on HiFi? Maybe to start, I'd like to highlight the monumental and foundational work of the Telomere to Telomere Consortium. As we all know, they released the first gapless human genome earlier this year. Building on that, the HPRC, the Human Pangenome Reference Consortium, which is kind of synonymous with T to T these days, has recently released a series of preprints describing the generation of high-quality diploid reference genomes and the first human pangenome reference. So in this series of papers, the authors used HiFi to generate the most complete, contiguous, accurate, haplotype-resolved, and cost-effective diploid genomes. And we're really looking forward to seeing what effect Revio can have on the next generation of research like this. From this work and from this experience, the HPRC has said somewhat of a provocative statement in that they said that we no longer consider collapsed 3 gigabase genome assemblies as state-of-the-art, but instead consider 2 genomes for every diploid genome assembled, 6 gigabase pairs versus 3 gigabase pairs, where parental haplotypes are phased and fully resolved. So it really takes into the account that each cell has 2 non-identical copies of the genome, one from mom, one from dad, and it's technologies like long-read sequencing that will push this as the new paradigm. So let's talk a little bit about 5MC, what are researchers doing with 5MC. So the value of 5-base HiFi sequencing was really well demonstrated in this preprint from Children's Mercy Research Institute in Kansas City. What they did in this study was they analyzed data sets of 276 samples from 152 families. The researchers found excellent concordance with whole genome bisulfite sequencing. However, the long reads allowed them to phase and discover that the hypermethylated sites were allele specific, so they were on one allele or the other. Additionally, the average number of hypermethylated sites that they found with long-read sequencing versus whole genome bisulfite sequencing was 117 versus 8 for WGBS. And then finally, the long-range sequencing, or the long reads, allowed them to link the genomic events and the hypermethylation effects of those genomic events. So they were actually able to do linkage across kilobase regions. So the previous two examples were where HiFi was mostly used alone, or with a couple other technologies. But it can be used as a standalone. However, HiFi is also very, very valuable as a complementary technology to short reads, and this is demonstrated very, very well by the All of Us program and the Broad Institute. Recently, just last September at AGBT Precision Health, they announced that they'd be sequencing 10,000 HiFi genomes as a complementary data set to their pre-existing short-read whole genome sequencing data sets. And they did that because the short reads are really, really good at resolving small variants, but they wanted to add data on top of a subset of those samples to get structural variant data, to get tandem repeat data, to get phasing, and to get methylation. And you can do that very economically with a shallow genome, 8 to 10x genomes. You may be wondering, what do I do with this long-read data if the variants that we're finding are novel? What do we do? How do we make sense of them? To that end, we have joined the Consortium for Long-Read Sequencing. We call it COLORS. The mission of COLORS is to form the COLORS database, which is a variant frequency database from long-read sequencing. And so the idea here is we want to help you do frequency filtering of these novel variants that you're going to find when you do long-read sequencing. Right now, we have about 2,000 genomes from 9 different institutions and growing. We are determining which standardized pipelines we'd like to process these genomes with. Once they are processed, we are going to upload them to NHGRI Anvil, and they're going to be available for your use, for the public use, so that you can, analyzing whole HiFi genomes will be easier for you. I have mentioned tandem repeats a few times, and that's because this year, our computational biologists at PacBio have developed a tandem repeat genotyping tool. So we have a tandem repeat genotyping tool. It's called Target, TRGT. The way it works is they have been collaborating, our informaticians have been collaborating with other scientists. They've developed a catalog of tandem repeats genome-wide, or catalogs, that the tandem repeat genotyper uses those catalogs and it genotypes the HiFi genomes. And in this example, the way it works is this is just one expansion. It's actually a Huntington's expansion, but you have the definition of the tandem repeat, right? You have the start and the stop. You have the motif, the CAG repeat, the CCG repeat. And then the tandem target then gives you out the repeat sequences, repeat lengths, methylation levels, interrupting sequences. And here's an example of the simplified visualizer that you get. You get the 2 haplotypes, you get the CAG repeats, and the numbers represented by these tick boxes, the CCG repeats, interrupting sequences, insertions, and deletions. If you wanted the methylation status, you would look at it in an IGV view. Let's not forget about RNA. In this paper, these researchers did single nucleus isoseq analysis in aging Down syndrome brains. They did a 10x prep. They split the material and they did half of it on short reads, they did the other half on long reads. And what they found was thousands of novel isoforms with the long reads that didn't exist in the short read data sets. And what they said was that these thousands of novel isoforms could be a great well of discovery in determining diseased and normal brains, or differentiating from diseased and normal brains. All right, so we have also targeted sequencing. I just want to talk about that really quick. 2 flavors. We have amplicon sequencing and target enrichment in partnership with Twist Biosciences. We have a growing body of literature to support you in both of these if you would like to do that with HiFi. Finally, this is my last slide. We have a dark regions panel that will be coming out in partnership with Twist. Fritz is really the designer of that. He's going to talk more about that. Just to give you a sense for the scale from Sequel 2e to Revio, on Sequel, conservatively, you can put 4 samples onto a SMRT Cell at a price of about $300 in sequencing reagents. The throughput increases on Revio will make that about $89 per sample. And with that, I'd like to introduce Fritz Sedlazeck from Baylor HGSC. He's an associate professor there, and he's going to talk to us about challenging medical relevant genes.
So today I'm talking about closing the gap, solving complex medical relevant regions on the human genome. And as I said, this is really a big collaboration of ours with Twist and PacBio itself. The motivation behind it is pretty straightforward, as you know, and might have seen this slide. Our understanding of structure variants or complex or repetitive regions is really growing with the technology at hand, being in the 1940s, 1980s with karyotyping, going really into genomic microarrays into the thousands, and then with Illumina sequencing coming around 2008, 2009, right, where we had the first really details on a base pair level. But we also discovered very rapidly that Illumina is sometimes kind of falling short of fully discovering these complex alleles and complex genes. And so there is really the single molecule sequencing from PacBio as well as Oxford Nanopore that is very intriguing. So our group at Baylor, as well as like many other groups across the time, have published multiple papers showing that we can better resolve structure variants as complex alleles than short reads. So here we can often discover 20, 23, 24,000 structure variants in a healthy individual, whereas with short reads, we often just get like around 12,000 structure variants per genome. It also allows us to access this repetitive region. So in 2019, Mendekler introduced, I think in 2016 actually, Mendekler introduced this 193 medical genes that are so repetitive that is often not accessible. And so we really need long reads. The T2T, as just mentioned, is also a big pusher in terms of repetitive and new regions of the genome to resolve them, which is just possible with PacBio and Oxford Nanopore. And then the other thing about not just assembly, but also phasing, meaning that we can identify which mutations are co-occurring on the same molecules or different molecules, is really an essential part, which I think is often overlooked. So a lot of these insights are actually pushed through genomic benchmarks that we have now available and that we worked with the Genome in a Bottle consortium with, being, for example, a structure variant benchmark that has been published in Nature Biotech, where we have sequence-resolved structure variants of large insertions and large deletions, which was derived from multiple technologies. However, the downside is just insertions and deletions, so like the really complex rearrangement events we don't have covered in this. And also, quite frankly, it also covers mainly boring regions that we all can kind of assess. On the other side, we have the SNV benchmark, including short indels, of course, that covers also more hard-to-assess regions such as the MHC. It's directly and more repeats. But the question that I always wanted to raise here, and actually we came to that first discussion at ASHG 2019, the last time we were all here in person in Houston, is like, what about challenging medical regions? We all know there is SMN1, 2, we all know that there is like LPA and these kind of regions that we really need to crack down for, right? And so together with Justin and his group, we worked on this challenging medical benchmark, where we took in a collection database of medical genes, compared them to what is already available in Genome in a Bottle in terms of benchmark. So those were a total of 5,000 medical relevant genes that we took in. Around 400 of them were not already covered by any benchmark in the genome. And so we would develop together with PacBio, based on an assembly, this kind of benchmark for medical genes. One of the interesting things that we actually found is that we also identified errors in the human reference genome of GRCH38, where regions are actually accidentally duplicated, and not just like random regions. Those are really medical important regions. And so the question is also coming up, how do we fix this? And so one of the things I briefly want to highlight, because a postdoc of mine is having a poster right now about this, is this little tool. So like, as I said, initially, so here are some short reads I'm showing. Initially, you cannot map in these medical relevant genes, but then like if you use his tool, you get the lower track from IGV, where you can see the mapping is much more robust. We actually can identify SNVs in here. So with that, we can improve the recall and precision of SNV calls. And then we also have collapsed regions that we want to improve and identify. So like if you're interested in how to fix these things rapidly, also in your short reads, this tool just takes 10 minutes. It's called Fix It Felix. We just changed the name. and the posters right now happening. So coming back to this medical gene benchmark, where I really want to talk about today. So as I said, this benchmark includes 386 genes that we tried to fully characterize. What is interesting is that 122 of these genes were too hard to fully characterize, or we really struggled with the variant representation in these genes. So a lot of problems here is the comparison of variants, but also some of the resolving part. What is interesting is here mapped out the contribution of all these 400 genes almost to different disease levels. You see most of them are actually 15% are neurological diseases. I didn't label any cancer genes, but there are also cancer genes in there like TB, TPEN, and other very interesting genes. And so with that benchmark in mind that we published earlier this year in Nature Biotech, we really kind of set out and thought like, what can we do with that? And how can we kind of go further, right? And so one example that gets close to my heart, so to speak, is LPA, which is a very important gene for cardiovascular disease risk. Just a very short story, LPA has a copy number variant, which is above the KIF2 region, which is a 5.5 kb region. And that is repeated like 5 to 50 times in every individual. And the number of copies is directly linked to your cardiovascular disease risk. And it's highly polymorphic across ethnicities as well as the general population. And getting this copy number right is really important for assessing your cardiovascular disease risk, but it's very hard to do that. And then like also resolving SNVs in there is very hard to do that. Nevertheless, the gene is highly important. For example, Washington Post put out an article about that. And we are looking into using graph genome approaches to resolve this region a little bit better and more. But like what this work really showed us is like we need really long reads for these like medical genes to get that right? And so like, how can we go kind of beyond, right? Long read sequencing is still a little bit expensive to do it on the whole genome every day, so to speak. So we need a routine assessment of these genes. For doing that, we need lab optimizations together with Twist. And then we also develop computational methods to go forward here. So as I said in the beginning, we partnered up with Twist and PacBio to really tackle this point. And the key idea basically right now is you can do your Illumina genome or existing data sets, and then you pay a few bucks extra to get long reads on these medical relevant genes around the 389 right now. The whole target design is a 22 MB, and it's really the nastiest and trickiest genes that there is in the human genome that have been shown to be relevant. So the idea is really to get the most bang for your buck, so to speak, in terms of long reads, having that cost investment. So how does we, of course, ran already this panel and designed this panel with Twist, and PacBio helped us to run this data. So how does the first result look like? Well, this is HG002. This is the genome in a bottle sun, where we can compare to the variant cause from our benchmark. So the on-target rate right now is 52.2%. This is still something in work, but this is also because these genes are so repetitive, right? So like we're not only capturing the genes per se, but we also actually interestingly capture the pseudogenes of these genes and can investigate not just the genes, but the pseudogenes and the interaction of them in terms of recombination events. Overall, you can see the coverage is very well. We have a few genes that we don't capture right now with this panel, but overall, most of the vast majority of the genes are very well covered. You see here a dashed line where the 20x coverage is indicated, and the percentage in terms of GC and HE content. There's a little bit of a GC bias in here, but like overall the data looks really stellar and really good. And the average coverage across all the genes is highlighted here again with a 20x cutoff as a dashed line, so you can see like 80% of all the genes are completely covered with 20x on the 4-plex design as Ed just mentioned. We also tested out an 8-plex run, and we're still evaluating the data set, but actually it looks pretty good. So like I think going forward, we can even halve the costs for that thing. So how does the variant calling look like? Here are the plots from a simple mapping approach and a genomic variant caller approach. It actually looks pretty good. Based on the HiFi data, we have a high F1 score, so that's the accuracy, basically a arithmetic mean between precision and sensitivity. And you can see that most of the genes we can call very well. Some of the genes we cannot. Some of the genes are zero coverage, as I said. Some of the genes, actually 7 of them, have no variants in gene on the bottle, so it's hard to kind of get the variant calling right. But the precision we get right, of course, in them. So the first data really look actually really good. However, I think we can improve above that. So on the recent data sets, on the recent lab optimizations, we had multiple runs in our center with that design, and you could see that over the runs we can actually get the on-target rate a little bit higher, which means that we have more efficient in capturing these genes and can more multiplex these samples together, bring down the costs on all of these samples. We also tried a PCR-free approach from Donner's group, and really trying to further optimize that in cost reduction. So the other point I want to highlight a little bit more is the methods to assess these genes, because I just showed you a simple SNV calling based on mapping, right? And the performance looked actually pretty good, but there are some genes like SMN1 and 2, or like LPA, where we actually need specific bioinformatic callers. And me being a bioinformatic person, I always like to talk about these a little bit more than the lab side. So here's, again, some IGV screenshots just showing you why we think this really matters, right? So for example, on the top here is GBA gene, which is a really important gene for MSA and Parkinson. You can see here on top our panel from the PacBio HiFi reads, on the bottom the Illumina reads right now. And you can see that part of this gene is mapped with Illumina, but like not comprehensively and not completely. And as I said, our panel then also includes the pseudogene, and we can investigate mutations in both of these copies of these genes. Here are some more tricky mutations that our panel can resolve. So here's the gene TERT, where you can see large insertions that we can resolve with our gene panel that otherwise are not being resolved at all. And here's the gene TYK2, again showing a 300 base pair, probably ALU insertion right in the intron here that otherwise are being misrepresented and misresolved. So what are we currently doing in terms of the analysis and mapping is, as I said, our preliminary analysis pipeline is including just the mapping and an SNV calling, and we are assessing these regions like that. We are currently putting on top of that the finished pipeline for phasing and SV calling, and then also having incorporation of assembly for certain genes. So here's, for example, my favorite gene LPA, where you can see how we are covering that in the panel itself. So we marked here the genes in 2 haplotypes because we can use these long reads to phase through the entire gene and really resolve both haplotypes, indicating different copy number variants and different mutations on each of the haplotype, which could have some impact on your disease risk. And then if you're interested in how we are doing structure variant calling further on long reads, Luis, my postdoc, is presenting Sniffles 2 also today, actually. But as I said, besides the mapping approaches, we also have to have like specialized tools, and together with PacBio, they developed multiple tools now. So Xiao from PacBio developed an SMN1 and 2 caller, where you can see here the SMN1 region. Again, on the top panel, you see our panel design. So gray reads meaning significantly uniquely mapped reads, and the white reads from Illumina indicating really hard to identify of SMN1 and SMN2, what are these reads. And her tool is able to kind of haplotype resolve not just SMN1 but SMN2, and gives you much more insight in these complex genes which have profound impacts. On the lower part, I show you a screenshot from CYP36. Again, the upper part is from our panel, lower part is from Illumina, just bringing down why we need this. And there's also a CYP36 tool or completely CYP family tool set developed from John from PacBio as well. And then, as I said, GPA as well as LPA are also very tricky genes, not just because of getting the copy numbers right, but also which kind of new mutations we can find and which new mutations can be phased. And so, for example, here we are showing a craft genome approach that we are currently implementing with them. Seth is mainly leading that from PacBio, where you can see all the different complex haplotypes and links of this KIF2 region that is multiple repeated. So you see here the copy number repeat as a kringle from that LPA thing. And this is all currently tested and currently implemented on top of our panel design. So making really a cost-efficient, I think, approach to target these genes specifically. So to kind of summarize what we have done and what we are doing, future steps is like we're really trying to further kind of squeeze a little bit the whole panel and make it more efficient in terms of scalable and cost-efficient method to assess these critical medical relevant genes, not just in terms of SNVs and SVs, but also in phasing, repeat expansion, and then specialized tools. We are currently putting together this analytical pipeline that you can not just like buy this panel and run it through the PacBio machine, but then also have a very easy approach for identifying these mutations in your samples itself. As I just introduced, there are multiple specialized tools that we're currently plugging together. And so I really think, and that's what gets me excited, is that this panel is really, I think, the first approach how we can have a large-scale application across multiple diseases about multiple individuals in this medical relevant challenging genes. And so like as Ed just introduced and PacBio introduced yesterday, we are already looking into the new sequencer, the Revio, and how that would help us to further scale and produce more cost-efficient data. So as Ed already showed, we are basically looking from a 4-plex now into the 12-plex in this system, which brings down the cost further in this panel. And we are investigating also higher plexes. So here is, for example, a screenshot that I got from PacBio yesterday. We also got the data from that yesterday across GPA against this really important Parkinson and MSA gene or neurological disease gene, where you can see that all these samples really have a high coverage with beautiful SNPs called. And so this is really something that we just got the data from, so we didn't have time to really run a lot of things, but this is something that Matt Head, my postdoc, and I are currently really looking forward to dig in and explore further how we can utilize this system. So with that, I want to say thank you for your attention, for coming here. I want to say thank you, of course, to my postdoc, Matt Head Mahmood, who just gave a talk also here at ASHG, and of course the entire center with Donna Ginger, Richard Harsher, Shalini Siad, and he. I also want to say thank you to PacBio for an awesome collaboration, especially Sarah and Geoff, who helped me kick this off, and then especially John and Xiao, with whom we have regular calls for improving the bioinformatics approaches. And then of course Holly, Tina from Twist Biosciences, with whom, like without them, it wouldn't have been possible to approach this and design this panel. And yeah, thank you very much for your attention. Before everyone goes, I'm also looking for postdocs and other people for joining my lab. So like, please ping me or reach out if you're interested in this kind of work. Thank you so much.